Good morning, everybody. This is Town Peterson welcoming you to the 18th Global Online Seminar in Biodiversity Informatics. I am very pleased to welcome our speaker this month, Enrique Martinez-Meyer. Enrique did his undergraduate and master's studies at the National Autonomous University of Mexico, and then came here to the University of Kansas for a PhD. He did his PhD in geography and has since then built quite an interesting program of studies of global change and how global change relates to biodiversity. He's now the equivalent of a full professor in the Instituto de Biologia in the National Autonomous University of Mexico. So Enrique will be talking to us today about essentially taking species distribution models or ecological niche models, whatever you want to call them, beyond simply estimating distributional areas and getting to the idea of the possibility of estimating abundances. I'm going to turn it over to Enrique in just a second. But I will remind you to send your questions as promptly as possible, if possible during the seminar, because I need to pass them on to Enrique at the end. So you can send those to biodivtraining@gmail.com or just to my email, town@ku.edu. Please do send questions so that we get some interchange with Enrique at the end of this talk. But with that, I'm going to turn it over to Enrique, and I will come back at the end to manage the questions. Thanks very much, Enrique, for accepting to give this seminar.
Hello, Town. Hello, everybody. Many thanks, Town, for this invitation. I really appreciate it. Today, I'm going to talk to you about these ideas that I have had for years now, for a decade ago, since I was a student at the University of Kansas, and these ideas were originally discussed with Town as my main advisor. But in the years, this has been evolved in different more comprehensive and more integrative ideas on how the theory of the niche and ecological niche modeling can explain and can help us to model the distribution of abundance across the distributional range of species. And let me start with the presentation right here. So the title of the presentation is like that: Going Beyond Distributions, Ecological Niche Modeling and Estimation of Species Abundances. And in these years, in this decade, many people have been involved in different ways to these ideas. My students, Daniel Diaz, Carolina Ureta, Carlos Yáñez, now Claudia Moreno and Patricia Martinez, but also other students like Berenice, Jiménez, Cintia Ureña, Constantino González, Ángela Cuervo, Andrés Arias, and my colleagues, Town Peterson, Octavio Rojas, Miguel Nakamura, and Jorge Soberón, are people with whom I have been working and discussing these ideas. So what I'm going to present you today is the product of all this thinking and exercises that we have done. Well, in the 80s and 90s, Professor Jim Brown from the University of New Mexico published this couple of papers.
Enrique, sorry to interrupt you, but you need to unshare and reshare your screen so that we can see your presentation.
Okay. Let me see what happened here. Sorry, Tom.
Okay. Unshare the screen And now reshare it, but selecting entire screen or all windows.
Yes.
Now you can go to PowerPoint and go into presentation mode.
Okay. Now you see the full presentation?
Yep, that's perfect.
Okay, thank you very much. So, reloaded. In the 80s and the 90s, Professor Jim Brown published these couple of papers in which he analyzed the internal structure of geographic ranges of species in terms of the abundance. And he found, or he described, three main patterns in terms of the distribution of abundance across the ranges of species. In the first place, he observed that for all species that he analyzed and from other species that he didn't personally analyze, but he looked at the information, he saw that in general, there are most of the places where species occur, the number of individuals are low. There are low densities, and very few sites have orders of magnitude more individuals. And this is a very general pattern from vertebrates to plants to microorganisms. So distribution of abundance is not homogeneous across the whole range. This is one of the patterns. The second one is that there is a strong spatial autocorrelation in abundance. In other words, nearby sites have more similar abundances than sites that are farther away. In the third place, he observed, and people before him, that highest abundances for several species were found towards the center of the geographic distributions. More finely viewed, this pattern is that higher and lower abundances can be found at the center of the distribution of species. And towards the edges of the distribution of the species, only low abundances were found. This last pattern is called the abundance center hypothesis. And it has been taken in biogeography and ecology almost as a dogma. In many studies, they take this like the rule without even testing if this is true or not. Speaking about this abundance center hypothesis, a formal testing of it was made by Rolf Zagarin and his colleagues, and he found that this is not a universal rule as it was claimed before. Actually, they found that in a meta-analysis that not even half of the information or the analysis or the studies they analyzed follow this pattern. So it's not a universal rule. It's something that sometimes happens and sometimes it doesn't happen. Well, what processes or mechanisms are behind these geographical patterns of the distribution of abundance? There are some hypotheses put on the table. For example, that the dispersal dynamics, population dispersal analysis of individuals are responsible for these patterns. This was proposed by Polyman. Then Brown himself proposed that it's more related to physiological tolerances, that population fitness is the response of the physiological response to one or more critical environmental variables for the species. And following these ideas of tolerances, Brown, in his 1995 paper, stated that this spatial variation of abundance is an expression or reflects how much local sites satisfy the niche requirements of species, and he referred to niche requirements, or he was thinking on the Hutchinsonian concept of the niche, this n-dimensional hypervolume that Hutchinson proposed as a niche. And well, here is where we, from these conceptual ideas, where we think that ecological niche modeling or ecological niche models maybe have something to say about the distribution of abundance. In these examples that you see on the screen, it's an ordinary Maxent run for data on lions, and as you can see, there are different shades from dark brown to light brown, and kind of intuitively, when we see this map, which in Maxent language is the probability of occurrence of lions in Africa, we can think that those dark areas represent highly suitable sites for lions, and therefore these areas may hold higher number of individuals. So it's like of common sense to think that areas that are more suitable for species may hold larger abundances. Well, in recent years, there have been several studies now that have tested the ability of different modeling algorithms to predict the abundance patterns across the species geographic ranges. This is probably the first one of this kind, from Jeremy van der Waal in Australia. He used Maxent to derive suitability in scores, and he tested if these suitability scores have a relationship with abundance via quantile regressions. He concluded, well, this study is very interesting. He concluded that yes, there is some information that Maxent rescue from abundances. There is a relationship, a positive relationship between suitability, environmental suitability, and abundance, but it's not linear. And the best thing he could get from Maxent is a prediction of the upper limits of abundance. If you see in this little box here, in the graph here, this is a very common pattern of the relationship between suitabilities and abundance. This is a triangular relationship in which high suitabilities may hold both high abundances and low abundances, whereas low suitabilities normally hold only low abundances, and makes this triangle. Jeremy and his colleagues proposed that this triangular relationship comes because in the highly suitable environments, you may have low and high abundances because there are other local factors that are not included in the modeling exercise that affect abundance. In terms of the theory of the niche or the niche theory language, there are other Eltonian variables which affect the abundance more than these Grinnellian variables that we normally use in niche modeling. Well, there are other studies with negative results. For example, this from Alberto Jimenez Valverde, in which he tested this relationship using a neural network for 48 species of arthropods, and he found that only in 9 of those species there was a positive relationship between probability of occurrence and abundance. In this other study from Natalia Torres in the group of Alex Diniz Filo in Brazil, they tested the ability of several, you can see here to the right, of several algorithms to predict the abundance of this species, the jaguar, across the whole distribution in the Americas. If you see here the R-squared scores and the p-values, they found that very few of these algorithms have a significant, statistically significant relationship with abundance, for predicting abundance. And if you see the values, they are quite low. The R-squares are quite low. Here is intriguing. There are two things that intrigues me from these studies. In the first place is what do output of these different algorithms really mean biologically. If you think on these results that I am showing to you, these are only statistical relationships between algorithms and abundance, but there is very few or none biology behind. I think all these studies are lacking like the theory, the ecological theory, to understand these relationships. And in this sense, there is theory that has been developed from old years. This is a study from Professor Bassett Maguire from the University of Texas in Austin. And following the original ideas of Hutchinson, of the niche concept of Hutchinson, Dr. Maguire thought that this hypervolume does have an internal structure. In other words, not all parts of this hypervolume of the niche of the species is equally good for the species. And he tested these ideas and observed that there are regions in this hypervolume in which birth rate is maximized and death rate is minimized. So abundance is higher in these regions, and these regions hold the most suitable or appropriate conditions for the species, the optimal conditions for the species. And as we move apart from this region, birth rate starts to reduce and death rate starts to increase. So abundance, in general, abundance decreases monotonically all the way to the edge of the ecological niche of this hypervolume. If we think on how species relate to environmental variables, there are many different ways in which species respond to environmental variables, but the simplest one is of this shape, of this Gaussian or bell-shaped relationship, in which we have lethal physiological tolerances, and then we have these spaces of suboptimal conditions, and in the center of this distribution of this curve, there are optimal conditions. If we extend this idea to a multidimensional space, we can construct this hypervolume or these Maguire ideas on optimal and suboptimal and marginal conditions. And optimal conditions should be around the centroid of this hypervolume. So we started to think on how to test these ideas. So we hypothesized that there should be an inverse relationship between the distance to the niche centroid and abundance in this multidimensional space. So with my first student working on these ideas, Daniel Diaz, we tested the existence of this relationship, inverse relationship between the distance to the niche centroid and abundance, using, in the first place, this species of bird to honor Joseph Grinnell, one of the founders of the niche concept. So what we needed to test these ideas was a bunch of data with abundance information across the whole distribution of the species. So we went to the BBS, the Breeding Bird Survey database, which has counts of birds across the distributional, across the distributional range of all species in North America and southern Canada for several decades now. Then we needed the niche dimensions or niche variables, so we used the word claim and other variables, and then we modeled the distribution, not the distribution, the ecological niche of the species using the standard techniques for modeling niches. In this case, we used GARB. This was about 2005. And under this approach, what we were trying to do was to reconstruct the hypervolume, the Hutchinsonian hypervolume here, using the environmental variables and presence records, occurrence records. We are not using any abundance information here. It's just we wanted to know what are the tolerance limits for the species, and then produce the potential distribution map, which is this one. From this map, we extracted the information of the environmental variables at each one of the pixels in which it was predicted presence, the species, and then calculated the niche centroid as the standardized mean of all environmental variables. And then we used the information from the species that we have data on abundance to calculate the distance of these points to the niche centroid, and then on using a regression analysis to see how if we could find a relationship between abundance and distance to the niche centroid. And this was our first result. We found a significant and pretty decent relationship between the distance to the centroid and the abundance. Then we compared this relationship in the geographic space too. We calculated the geographic centroid and then related it to abundance, and we found very little or a very weak relationship between these two variables. And also we modeled the distribution of the California threshold with Maxent, and regressed the output of Maxent, the probability of occurrence, against abundance, and we found basically no relationship. So in other words, Maxent was unable to predict the abundance of Toxostoma. This was quite an exciting first result, so we continue to do this analysis with several other species of birds from the with data from the breeding bird survey, using the distance to the niche centroid, the distance to the geographic centroid, and the maxent output. And you will see in the following slides, I want you to see the strength of the relationship in these three analyses, and you will see that in all cases there is a stronger relationship, or there is more explanation on the dispersal of the data using the distance to the niche centroid than the geographic centroid or the max censorability scores. I want you to notice two things. First one, here in the distance to the niche centroid, there is an inverse but not linear relationship. There's always this shape of the curve, and there is also a lot of dispersal of the data, mainly in areas near the centroid, or what we call the optimal areas. Here. And this is constant across all the species that we analyzed. This is another bird species. Also, you can see the distribution of the species. And notice that for those species that have fairly circular distribution, the relationship between the distance to the geographic center and abundance is higher. Then we look for another type of information, for data for different species other than birds from the breeding bird survey. And Daniel found information from other species, in this case like the wolf. And this relationship holds across other species, in this case mammals, even a tortoise, and other species in different regions, not only in the Americas, but in Africa and Asia, etc. So we were very happy with these results. We think that the response of populations in geography is explained mainly by the distribution of the populations in the ecological space, and the expression in the geography is what produces these geographical patterns of abundance. Now, why is this relationship important? Well, if this relationship between the distance to the niche centroid and abundance holds across different species, different taxonomic groups, then we have a tool to map the distribution of abundance using only presence data. This is, in this slide, you can see the distribution in this map on the top left. You can see the distribution of abundance modeled with presence data. And in these relationships here, down, you can see that there is a strong relationship between observed abundance, independent data of abundance, against our modeled abundance. We have done further testing, going to the field and getting information on density of different species. One of the persons who has been more active in these ideas is Carlos Yáñez, a former PhD student of mine, and currently he's a professor at the National University of Mexico. He went to the field and collected data on the density of the white-tailed deer in a small area, and two small areas in Mexico. The white-tailed deer is a species with a very, very large distributional range from southern Canada to northern South America. So he just went to two sites, estimated the density of deer, and then calculated or modeled the abundance under this distance to the niche centroid approach, with very interesting and successful results. He found that a map produced with only the distances to the niche centroid really resembles the distribution of abundance. So in other words, if we don't have any abundance information to test the relationship between abundance and distance to the niche centroid, we have some confidence that mapping the distance to the niche centroid is a good proxy for relative abundance, at least. And this is another study from with the land tortoise, the Mapimi tortoise, made by another master student, Cynthia Ureña. And in this study, we learned another lesson. Our first exercises of this kind, we were calculating the distances to the centroid of the niche using Euclidean distances. In this case, we, instead of using Euclidean distances, we used Mahalanobis distances, because it seems more appropriate, because Mahalanobis distances take into account the covariance among variables, and because several of our environmental variables that we use for modeling the niche of the species are correlated, there is a problem of collinearity there. So Mahalanobis distances take into account this effect. So what are the main lessons that we have learned from all these exercises? First is that the spatial variation of abundance is better explained by the niche, the internal structure of the niche, than by the geographical structure of data. So we firmly believe that when abundance is an expression of population fitness, that not in all cases this happens, then the analysis of the internal structure of the niche can give us information about the geographic patterns of abundance. Second, the relationship between the distance to the niche centroid and abundance is better described by the simplest distributional function, which is a multivariate normal distribution. With a friend and colleague of mine, Miguel Nakamura, from the Math Center, a math research center here in Mexico, he ran some simulations with hypothetical data to test which family of distributions best reconstruct the empirical data. And he found that this multivariate normal distribution is the family of distributions that better describes the empirical data. This is true when the relationship between, or the response curve of the species to the environmental variables follow this Gaussian bell-shaped distribution. But there are instances in which the optimal for the species is not towards the mean or the center of the environmental gradient. Think, for example, in extreme or extremophile species, like those that live under, for example, full aridity, in those like Atacama Desert in South America, in which there is no humidity for years. And in a humidity gradient, of course, species wouldn't, or species adapted to those environments would not respond to this humidity gradient in a bell-shaped form. There are instances in which there is a saturation in the response of the species to the environmental variable. So when this happens, when the relationship between the abundance of species and the variable is not bell-shaped, this is what causes the dispersion of data that you see in the regression analysis. Let me go back to show you where. Like in this case, for example, you see a lot of dispersion in data here. And this is probably due to the way the species interact with the environmental variables that is not in this uniform unimodal way. In another paper led by Carlos Yáñez, he also observed that sample size and sample bias also causes dispersion of the relationship, of dispersal of the data, and in the relationship between the distance to the niche and abundance. He used, in this case, a virtual species to test how different sampling biases and the data incompleteness affect the relationship. And we observed that the more biased the data or the more incomplete the data, the more dispersal you will find. So now we are still working on these ideas, both in the implementation and also in the conceptual part of these ideas, trying to understand how the theory of the niche and thinking on the internal structure of the hypervolume can explain the distributional patterns of abundance of species. But abundance, as I told you before, may or may not be an expression of fitness of the species. There are examples in which, well, let's put the simplest scenario. In sites where optimal conditions for the species prevail, as stated by Maguire, you would expect more individuals because there is more reproduction and birth than death. But there are some systems in which this doesn't work like that. For example, in mammal species, very territorial mammal species, optimal conditions may hold very few individuals because they are territorial and they chase away other individuals. So those optimal conditions may not hold the larger abundances. In another case that we are just analyzing now, with cycads, I don't know how to say it. In this particular species of cycad, we observed that abundance is not a good proxy for fitness. Instead, there are some morphological traits like size of the leaves, functional traits that do relate to fitness. And we found an inverse relationship between the distance to the niche centroid and these morphological traits. This is very interesting. One of my students, Carolina Oreta, she's interested in climate change and corn in Mexico, and she tested the idea of niche centrality, but instead of using abundance as a proxy for fitness, she used corn yields, and she found for some maize or corn races this inverse relationship very quick, because our data on yields is not very good. Also, my students and former students are working in developing and polishing scripts for our program to make this process much more easier. Also, we are working on the transferability of now these abundance models in climate change scenarios, because I'm interested in responses of species to environmental change and specifically to climate change. So we are now exploring the ability of this approach, the distance to the niche centroid, to model the abundance under climate change scenarios. And I think that's it from my part. Thank you very much. And if you have any questions, I will be very happy to answer them.
Thanks, Enrique. If you want, share your screen so that people can see your beautiful face. People, if you have any questions, as you know, here is the email address to which you can send them. Enrique, you and I'll throw a question at you just in case questions are going to come in.
Mm-hmm. Yes.
You know the question I'm going to ask you, because we've argued about this a lot.
Yeah, I can imagine. What are you?
You want to ask the question for me?
If there is one if there is one optimal, or the centroid is the optimal, or there are multiple optimal points in the multi-dimensional niche, something like that?
No, I mean, I kind of do believe in the idea of a single convex niche, and I'm intrigued by the possibility that the central parts of it would have higher abundances of species than the edges of it. That to me makes quite a bit of sense. My question is more, what do you do about what we would call a Wallacean species, which is to say a species that is not exploring out to the limit of its environmental tolerances simply by dispersal? Maybe there's some big dispersal barrier. Or maybe there's a sister species just on the other side of the river or just on the other side of the mountain range. How do you deal with those species that are not limited by their niche dimensions, but rather by barriers?
Well, we already faced that situation with real data from a cacti in the Baja California Peninsula. We went to get abundance data on 3 species of plants across the whole Baja California Peninsula. These species are endemic or quasi-endemic to this region. And if you see the shape of Baja California Peninsula, which is a long strip of land, we found for this species that is called, the vernacular name is called cardo, that towards the tip of the peninsula we found the highest abundances. And I'm sure that if the peninsula was longer, we would find higher abundances south of the tip of the peninsula. But actually, the land ends the distribution of the species.
So if you have a situation like that, isn't your niche centroid biased because you're maybe only seeing half of the tolerance range?
It's biased, and the approach doesn't help to predict the abundance of the species. That's the case for this species. We have very, very good data because this is a species that is very easy to find. It's very easy to count because it's a columnar cacti in a very short desert, so it's very conspicuous. So it's very easy to find them and to count them. That's why we selected that. So there is very, very little bias in our data, and we traveled the whole peninsula, all across and all along. So it's a very, very nice data set. And our approach of the distance to the niche didn't work with this species. We first thought that probably we were missing some important variables like edaphic variables, but we tested this idea with another species from the peninsula, and it worked pretty well. But this other species is an endemic species to the center of the peninsula. So it's clearly that this is a Wallacean case. So in this case, yes, probably the centroid of the niche doesn't exist in the geography. So the whole analysis is biased, and there is very, very poor predictive power.
But you should be able to pick out the species that are Hutchinsonian in nature, and
yes,
those should be the species where this works.
Yes, that's right. Yes, that's true.
Interesting. Thank you.
Mm-hmm.
I'm not seeing questions coming in. Usually, as soon as we end these events, questions come roaring in. So if you don't mind a couple of questions that come in that you might get by email or on the event page, everybody would appreciate your answers. So I guess I'll thank you very much for your time and for your contribution. Very interesting. You know that I'm intrigued by this work, so I should give everybody a disclaimer that when Enrique was here as my student, I told him that this area of research was just going to waste his time and not going to yield any fruit. So I'll apologize to you publicly right now. But no, more generally, thanks. This is potentially a very important area that you and your students and colleagues are opening up. So this is really neat. More broadly, everybody, this is number 18. Number 19 will be on Thanksgiving Day, which is to say last Thursday in November. And I will be announcing shortly our speaker. I think I have the person lined up. And then we'll take a break because the last Thursday in December is a little too close to Christmas. Anyhow, thanks again, Enrique, and thank you everybody for tuning in. And we'll see you next month.
Thank you very much, Town. Thank you, everybody. See you.